So in this video we are going to talk about apoptosis, which is a form of programmed cell death. So in apoptosis, the death of the cell is induced by activation of some intrinsic enzymes which breaks down the cellular DNA. Now, what are those intrinsic enzymes? They are caspases. So now the caspases derived their name because they contain cysteine in their active site and they cleave after the aspartate residues and hence the name caspases. Now these enzymes are present in all the cells but present in an inactive form and whenever the cellular damage occurs or the survival signals or growth factors of the cell they are lost. These caspases are activated and produce apoptosis, which we will see. Now, apoptosis includes two phases. First is the initiation phase, where the caspases are stimulated and a cascade of reaction occurs, where the initiator caspase activated activates other caspases as well. And finally, the terminal caspase gets activated and causes the cellular fragmentation. Now, this apoptosis has two pathways. First is the intrinsic pathway or the mitochondrial pathway, which is the most common pathway occurring in physiological as well as pathological conditions, and the other is extrinsic or the death receptor initiated pathway. So, let's start with the intrinsic pathway or the mitochondrial pathway. First of all, there are certain proteins in the cell known as BCL2 family proteins. These BCL2 family proteins are overexpressed in B cell lymphomas and hence they derive their name. Now, the BCL2 family includes some anti-apoptotic proteins that resist or prevent apoptosis, some pro-apoptotic fat proteins, and some regulator or the sensor proteins. So, the anti-apoptotic proteins include BCL2, BCLXL, and MLC1. And the pro-apoptotic protein include BAX and BAC, and the regulator or the sensor protein includes BAD, BIM, BID, PUMA, and NOXA. Now these anti-apoptotic protein contain 4 BH domains. That means they have all the BH domains from 1 to 4, whereas the pro-apoptotic proteins have only 3 BH domains from BH 1 to 3, and the regulator or the sensor molecules have a single BH domain only, and that is BH3 domain, and hence these proteins are called as BH3 only proteins. Now, the activity of these BH3 only proteins can be modulated by any stress or the damage to the cells, and hence they act as sensors. These anti-apoptotic proteins, these are present in the outer mitochondrial membrane and makes it impermeable and thereby prevents apoptosis. Whereas the pro-apoptotic proteins, which include BAX and BAC, on stimulation, these proteins oligomerize and form channels in the outer mitochondrial membrane, which allows cytochrome C to leak out. And this cytochrome C, along with various factors, causes apoptosis. So the diagram shows that in a viable cell, whenever the survival signals or the growth factors are present, they causes the production of anti-apoptotic proteins which prevent the apoptosis, and hence the cytochrome C is maintained within the intermembrane space, and the cell is viable. In an apoptotic cell, either there is a lack of survival signals or growth factors, or there could be damage in the form of irradiation, free radicals, and etc. So these signals would be sensed by our BH3 only proteins, and they will antagonize or oppose the action of BCL2, that means the anti-apoptotic proteins, and cause activation of the pro-apoptotic proteins back and backs, and they will oligomerize and form channels in the mitochondrial membrane, which will allow the cytochrome C to leak out. Now, what happens is this cytochrome C binds to APAF1, that is apoptosis activating factor, to form an apoptosome. And this apoptosome activates caspase 9. This caspase 9 is an initiator caspase that will cause the cascade of the caspase activation, and multiple caspases will be activated, and ultimately the caspase 3 will get activated, which is our executioner caspase, and it will perform the cellular fragmentation. Since the events in the execution phase are similar in the intrinsic as well as extrinsic pathway, we can discuss them later. Now, one more important point, that there are some mitochondrial proteins known as MAC or Diablo, which are released into the cytoplasm, and they bind to the cytoplasmic proteins, and they act as physiological inhibitors of apoptosis. They prevent the inappropriate activation of caspases and keeps the cell in alive form. So this was all about the intrinsic pathway of apoptosis. We will discuss the extrinsic pathway as well as the events in the execution phase in the next video. Thank you.